Dear student, I am Dr. Prakash Mungli, Professor of Biochemistry. So in this video, I am going to explain you oxidation of branched chain fatty acids, omega oxidation of fatty acid and oxidation of unsaturated fatty acid. Now let's get into the details. Oxidation of branched chain fatty acids. An example for branched chain fatty acid is a phytanic acid, and this phytanic acid, it comes from a fermented product of a plant material in a ruminant animal's gut. When a plant material is fermented, especially the chlorophyll, when it is fermented, there will be release of a phytol, and this phytol is further fermented into phytanic acid. And this phytanic acid, it is a multi-methylated 20-carbon fatty acid, and humans get this 20-carbon multi-methylated phytanic acid via consumption of dairy products, ruminant animal fats and certain fish. And you are seeing here the structure of phytanic acid, and it's a multi-methylated fatty acid, so the methyl groups are shown in the structure. And when we number the carbons here, so the carboxyl carbon is number 1 carbon, and then the number 2 carbon and number 3 carbon. And when you re-number this using alpha and beta numbering, so second carbon is alpha carbon and the third carbon is beta carbon. Since in the beta carbon a methyl group is present, so that's why a beta oxidation do not go on here, because there is a hindrance by the methyl group. So how to oxidize this type of fatty acid when beta oxidation is not possible? So the answer to that is alpha oxidation process. And the alpha oxidation will be going on in the peroxisomes, because peroxisomes, they will express an enzyme called phytanic acid hydroxylase. Name of the enzyme here is phytanic acid hydroxylase enzyme. As the name says, it is going to introduce hydroxyl group at the alpha carbon of a phytanic acid, that is the second carbon. By introducing hydroxyl group there, it is going to lead to or initiate alpha oxidation, and at the end of alpha oxidation, a bond between first and carbon will break open and it will be released as carbon dioxide. So there will be release of carbon dioxide at the end of alpha oxidation of phytanic acid, which will be going on in the peroxisome. Now what happens? So the 20 carbon phytanic acid is now becoming 19 carbon pristanic acid. So alpha oxidation is going to convert 20 carbon phytanic acid into 19 carbon pristanic acid, and with that we need to renumber the carbons now. So I am going to do that. So we have released carbon dioxide already, so first carbon is released as carbon dioxide, so I am going to take that out. So rewrite the fatty acid, so we have carboxyl group right here now. So numbering, start from the carboxyl group. So first carbon, number 1 carbon, second carbon and the third carbon. And the second carbon, as you know, is alpha carbon, and the third carbon is beta carbon. As you see, now in the beta carbon there is no hindrance, no methyl group. Beta carbon is free. A beta oxidation spiral can go on. Number 1, 2, 3 reactions that we have seen in beta oxidation of fatty acids will go on, and that will break a bond between second and third carbon, and it is going to release a 3 carbon fatty acid, and that is called a propionyl-CoA. So there will be release of propionyl-CoA, and with that release of propionyl-CoA, so we need to rewrite the fatty acid now, because 3 carbons are removed now. So I am going to take those 3 carbons from this fatty acid. So 3 carbons removed, so far 3 carbons propionyl-CoA and 1 carbon carbon dioxide removed now. So total 4 carbons removed from a 20 carbon fatty acid. Okay, so carbon dioxide, carboxyl group is here. So that's number 1 carbon, number 2 and number 3 carbon. Number 2 is alpha carbon, number 3 is beta carbon. So there is no issue with the beta oxidation process. Beta carbon is free. So beta oxidation will go on and it will be released as, this 2 carbon will be released as acetyl-CoA. So acetyl-CoA will be released now. Okay. So since we have released acetyl-CoA by conducting a beta oxidation, so I am going to remove that. I am going to take this out now, and also I am going to rewrite the fatty acid. So we have carboxyl group now. So we are going to renumber. So first carbon and the second carbon and the third carbon. Now the second carbon is alpha carbon, third carbon is beta carbon. So there is no problem with the beta oxidation now. So beta oxidation will go on, and this 3 carbon now will be removed as propionyl-CoA. So I am going to add one more propionyl-CoA to the list. So now we have 2 propionyl-CoA there. Okay. So since we have released a propionyl-CoA, so I am going to take this out now from the figure. So this is removed. And I am going to rewrite the figure. So now we have COO minus. So this is number 1 carbon, number 2, number 3. Now let's count how many carbons are there now. 3, 4, 5, 6, 7, 8, 9, 10, 11. So 11 carbon fatty acid is there now. So second carbon is alpha carbon, just like what we have seen before. Third carbon is beta carbon. No problem with the beta oxidation. So beta oxidation will go on. So at the end of beta oxidation, now we are going to release these 2 carbons as acetyl-CoA. So we have 2 acetyl-CoAs now. Okay. So now let's remove it from the structure. So these 2 carbons I am going to remove and rewrite the fatty acid with the carboxyl group COO minus and give the renumbering 1, 2, 3. Okay. Then we have 4, 5, 6, 7, 8, 9. So second carbon is alpha carbon, third carbon is beta carbon. So no problem with the beta oxidation. So beta oxidation will go on, and these 3 carbon will be released as propionyl-CoA. So I am going to add number there. So it becomes 3 propionyl-CoA now. So let's take this out from the structure. So I am going to remove all that. So what is now rewrite the fatty acid? It is COO minus is here now. Number the carbons 1, 2, 3, 4, 5 and 6 carbon. Now this 6 carbon will move into mitochondria. Actually this will go into mitochondria. Now the beta oxidation will continue in mitochondria. So that means branched chain fatty acid oxidation will go on in 2 subcellular organelles. One is in the peroxisome and other is in the mitochondrial matrix. So in the mitochondria what happens? Same beta oxidation will go on. So we have alpha and beta carbon. So beta oxidation will go on. The bond between alpha and beta carbon will break and will be released as acetyl-CoA, 2 carbon acetyl-CoA. So now let's add that number here. So 3 acetyl-CoAs. Okay. So now let's take this out from the figure. I'm going to take these 2 carbon out now. So 3 acetyl-CoAs released so far and 3 propionyl-CoAs are released. So 3 acetyl-CoAs and 3 propionyl-CoAs are released. Let me just rewrite the figure here. Now we have this carboxyl group, which is the number 1 carbon and then number 2 carbon and number 3 carbon and number 4 carbon. 1, 2, 3, 4 carbon fatty acid is now remaining there, and this 4 carbon fatty acid is referred as iso, and it will be in a CoA form. It is C double bond O S C O A, and that is the CoA form of a fatty acid. C double bond O S C O A, that is an activated fatty acid. it is called as 𝐒𝐮𝐴 molecule. So this 𝐒𝐮𝐴 is now released. So at the end of all this, so what happened is, so we have a carbon dioxide, 3-propanenyl-CoA, 3-acetyl-CoA and 1-isobutyryl-CoA. These are the products of oxidation of branched chain fatty acid, that is phytanic acid. So carbon dioxide, 3-propanenyl-CoA, 3-acetyl-CoA and 1-isobutyryl-CoA. Now propanenyl-CoA, we know that propanenyl-CoA go into methyl-malonyl-CoA. Methyl-malonyl-CoA is converted to succinyl-CoA. I have already explained you that in oxidation of odd number carbon fatty acid. Acetyl-CoA, you know, it will go into TCA cycle. Now what happens to the isobutyryl-CoA? Isobutyryl-CoA will be converted into propionyl-CoA and that will release carbon dioxide. Okay, so that's the fate of isobutyryl-CoA. Once you get propionyl-CoA, so propionyl-CoA, it will be converted into methyl-malonyl-CoA and then into succinyl-CoA, going into TCA cycle. So this is what happens to the products of branched chain fatty acids. So when the question is asked what are the products of branched chain fatty acids, there are two ways of putting it. One is, you can say 1 carbon dioxide, 3-propanenyl-CoA, 3-acetyl-CoA and 1-isobutyryl-CoA. That's one way of saying it. Second is, you can say it as 2 carbon dioxide, 4-propanenyl-CoA and 3-acetyl-CoA. How? Because you see, initially there is I have written carbon dioxide here, 1 carbon dioxide, and isobutyryl-CoA converted into propanenyl-CoA and another carbon dioxide released. So we have 2 carbon dioxide there and 3-propanenyl-CoA here and 1-propanenyl-CoA here coming from isobutyryl-CoA. So that means 4-propanenyl-CoA and 3-acetyl-CoA. So like this also we can say. So the products of branched chain fatty acid, phytanic acid, it can be said in two ways. One is 1 carbon dioxide, 3-acetyl-CoA, 3-propanenyl-CoA and 1-isobutyryl-CoA. Or you can say 2 carbon dioxide, 3-acetyl-CoA and 4-propanenyl-CoA. Okay. So that's about how a branched chain fatty acid, phytanic acid, is oxidized in 2 subcellular organelles. One is in the peroxisome, other is in the mitochondrial matrix. So in the peroxisome, phytanic acid is converted into pristanic acid, 19 carbon, by release of 1 carbon dioxide. This is done by α-oxidation, and the important enzyme in the α-oxidation is phytanic acid hydroxylase. And the pristanic acid undergoes a regular beta-oxidation process until it comes to 8 carbon or below. And then it moves to the mitochondria where further beta-oxidation will go on. So at the end of oxidation, both in the peroxisome and in the mitochondrial matrix, we are going to get 1 carbon dioxide, 3-propanenyl-CoA, 3-acetyl-CoA and 1-isobutyryl-CoA. And if you convert an isobutyryl-CoA into carbon dioxide and propanenyl-CoA, so we are adding 1 carbon dioxide additional and 1 propanenyl-CoA additionally. And this is what is the oxidation of branched chain fatty acid. So all that I have written here. So alpha-oxidation converts phytanic acid into pristanic acid. Phytanic acid hydroxylase initiates alpha-oxidation. Regular beta-oxidation will continue after alpha-oxidation until 8 carbon length, and then beta-oxidation will continue in the mitochondria. And so peroxisomal beta-oxidation releases alternatively 3-propanenyl-CoA, both peroxisomal and mitochondrial beta-oxidation, I would say like that, will release alternatively 3-propanenyl-CoA, 3-acetyl-CoA, 1-isobutyryl-CoA, and in the beginning there will be release of carbon dioxide. Okay, those are the products of phytanic acid oxidation. Now let's move on to see one of the disorder that is associated with a defect in the oxidation of phytanic acid. The name of the disease is Refsum disease. Refsum disease is because of a mutation in a gene coding for PHYH gene, and this PHYH gene is coding for phytanic acid hydroxylase. So here in this particular disease, there will be deficiency of enzyme phytanic acid hydroxylase. Now you know the importance of phytanic acid hydroxylase. So it is involved in the initial alpha oxidation of phytanic acid in the peroxisomal matrix. So there will be impaired alpha oxidation of phytanic acid in this disease. So what will happen? Phytanic acid is not oxidized, means phytanic acid will build up in the tissue, and the phytanic acid derivatives will also build up in the tissues and plasma. So it's a neurocutaneous disorder where severe signs and symptoms are related to brain, eyes and nerves. So cerebellar ataxia and retinitis pigmentosa, chronic polyneuropathy can be seen. Patients will have vision loss, and anosmia is present in almost everyone with Rebsam disease, that is loss of smell. And also 1/3 of the Rebsam disease patients will show bone abnormalities of hands and feet, and usually it is an additional finger that can be seen in the hands and feet, and also ichthyosis symptoms, especially hyperkeratosis of the palms and sole, can be seen in Rebsam disease. Treatment is low phytanic acid diet, that is restriction of dairy products, and avoid consumption of ruminant meat, which will avoid entry of ruminant fat into a human body. Okay, so this is what is all about Rebsam disease. Coming to our next type of fatty acid oxidation is omega oxidation of fatty acid. Now the omega oxidation, as the name says, so in a fatty acid there is a carboxyl carbon and omega carbon, or the methyl carbon. So the carboxyl carbon is number 1 carbon, and number 2 and all that as we move on. So we have alpha, beta, gamma. Like that, the last carbon, the methyl end carbon in a fatty acid, we call it as a omega carbon. So the methyl end here will be oxidized into carboxyl carbon. So basically methyl group will be oxidized into carboxyl group, as it is shown in the figure. As you see, methyl group is converted into carboxylic group. So once that happens, so at the end there will be a organic molecule where it will have 2 carboxylic groups on either side. Right side there is a carboxyl group, left side also there is a carboxyl group. That's why the product of omega oxidation is dicarboxylic acid. And where exactly this happens and when you are going to see omega oxidation? So omega oxidation, it occurs in a smooth endoplasmic reticulum, and that is catalyzed by a cytochrome P450 monooxygenase system of enzymes. So here omega carbon undergoes oxidation to carboxylic group to yield dicarboxylic acid, and these dicarboxylic acids will undergo a regular beta oxidation on either end and release acetyl-CoA molecules. Okay, so when you are going to see increase in omega oxidation process, so whenever a beta oxidation is defective, or whenever there is excess fatty acids that are coming in to the cytoplasm and not being able to be oxidized in the mitochondrial matrix, and also medium chain fatty acids that are coming from adipose tissue lipolysis. So these fatty acids, whether it is a long chain fatty acids not being oxidized in the mitochondria, which are coming into the cytoplasm, diverted into endoplasmic reticulum, or if it is a medium chain fatty acids, these are the molecules that will undergo omega oxidation, and that will give rise to increased levels of dicarboxylic acid. So that is why, remember, defect in beta oxidation will lead to appearance of dicarboxylic acids in the urine. So that is why I have written here, defect in beta oxidation may lead to dicarboxylic acid aciduria, means presence of dicarboxylic acids in the urine sample. So increased levels of dicarboxylic acids, it is an indirect indication of defect in beta oxidation or saturation in the beta oxidation. Remember that. That's the importance of omega oxidation of fatty acid. Coming to oxidation of unsaturated fatty acid. So all this time what we have seen is oxidation of a saturated fatty acid without having the fatty acids which do not have a double bond. Now unsaturated fatty acids, they differ from saturated fatty acids having a double bond. So how these fatty acids are oxidized? So the oxidation of unsaturated fatty acids, it proceeds in the same way as a beta oxidation of saturated fatty acids until a double bond appears. Until the double bond, it is the regular beta oxidation that is going on, and when the double bond appears, things will change. So what happens? So that means a double bond need to be reduced into a single bond like saturated. Unsaturation has to be converted to saturated. Unsaturated fatty acid has to be converted to saturated fatty acid. A double bond need to be removed, and that will be done by certain additional enzymes. So certain additional enzymes are needed here in the oxidation of unsaturated fatty acids. What are those additional enzymes that are required to remove the double bond? So you need to remember the names of these additional enzymes to remove a double bond, and those are cis-trans-enoyl-CoA isomerase, acyl-CoA dehydrogenase, di-enoyl-CoA reductase. These are the three enzymes that will remove a double bond and allow beta oxidation to continue. This is what you need to remember here. So cis-trans-enoyl-CoA isomerase, acyl-CoA dehydrogenase, di-enoyl-CoA reductase. So these are the three enzymes that will remove double bond and allow beta oxidation to continue. Now let me show you here in this particular figure, so how exactly oxidation of unsaturated fatty acid is going on. So using those three enzymes, that is enoyl-CoA isomerase, acyl-CoA dehydrogenase and di-enoyl-CoA reductase enzyme. So these three enzymes, they are going to reduce a double bond that is present at the 9th carbon atom, and after reducing the double bond, so regular beta oxidations will go on and a fatty acid is oxidized into acetyl-CoA molecules. So basically all we need to remember here is in the oxidation of unsaturated fatty acid is wherever the double bond comes in, those double bonds need to be reduced so that saturated fatty acid is made, and those double bonds are reduced by enoyl-CoA isomerase, acyl-CoA dehydrogenase and di-enoyl-CoA reductase enzyme. So that's how oxidation of unsaturated fatty acid will go on in brief. So this is all about oxidation of branched chain fatty acid, omega oxidation and oxidation of unsaturated fatty acids. Thank you.